Hi, my name is Maximos Goumas, and I'm a PhD candidate at Florida Tech. And today I'm going to be presenting on my current research, ongoing research on bioenergetic modeling of methanogens in Europa's subsurface ocean environment. And also I'll be looking back and forth because I have another monitor over here with a couple notes on it. But first thing, I'm going to be going over some overview and motivation of this current project. So the central objective of this project, as it says here, is to study the subsurface environment of Europa with the goal of predicting possible amounts of biomass and potential biosignatures. And then we plan to expand this to explore other worlds and analyze methanogens on other worlds. We also are collecting properties of these ecosystems from a bioenergetic standpoint to interpret future collected data from upcoming missions like Europa Clipper and make theoretical predictions of what those missions should see once they get to their locations. And then lastly, we also are going to be doing these simulations of these interactions of organisms and specifically simulating these methanogens with each other and the environment to determine the habitability and survivability of these environments, again, from a bioenergetic standpoint. So firstly, why do we want to look at methanogens? Well, first, a little bit of background in case anybody needs a refresher. Methanogens are these little microorganisms that produce methane. And specifically, we're looking at hydrogenotrophic methanogens, which consume hydrogen gas and carbon dioxide and produce methane. There are multiple lines of evidence that point to their survival in non-Earth environments. Specifically, a lot of studies have been done with methanogens on simulated Mars conditions or Enceladus conditions, and they show very high resilience to changes in pressure, changes in water content, temperature, pH, various different parameters of these environments. We also think that the necessary reactants for methanogens to exist and survive and grow are known to or thought to exist on Europa in either the ocean or on the surface itself. And these sort of microbes, methanogens and other microbes, are known to exist around hydrothermal vents on Earth. And the ocean floor of Europa is also thought to have these hydrothermal vents, which provide a source of energy or heat for these methanogens to exist. Also, we are looking at Europa, and why Europa? Well, it's thought that there's a large subsurface ocean, water ocean, and we know that water is required for life as we know it on Earth to exist and to grow and thrive. Like I said before, theoretically, it includes these hydrothermal vents, which we know have microbes surrounding them on Earth. So we would assume that the same would be the case on this moon. The presence of this source of energy or heat combined with the liquid water and protection from an icy outer shell provides like a perfect environment for these microbes to exist beneath the ice shell. And lastly, like I mentioned before, there's a mission Europa Clipper is coming up, and it's going to be looking at Europa and looking at various chemical composition of the surface and the atmosphere, and potentially any gases that can come up through the ice from the ocean below. And we'll be able to use our research to help predict what Europa Clipper should see. So, and here's an image of Europa Clipper as well. To do these simulations, we'll be using a code called Nutmeg. It's a publicly available code written by Peter Higgins and Charles Cockle. The Nutmeg name stands for Nutrients, Maintenance, Energy, and Growth. And the authors describe this code as an astrobiological model for assessing the energetic and nutrient availability of environments to predict their potential biological productivity. And it can be used to estimate how much biomass an environment could host and how that life might affect the local chemistry in that environment. And why do we choose Nutmeg? There are not many models available that can do this. The structure and the methods used in Nutmeg allow for a lower computational cost, and thus I can run it on my own personal computer, and the simulations don't take very long. So far the simulations I've done have been on the order of 3 to 5 hours at the longest. And also Nutmeg offers a very vast amount of customizability and plenty of room for expansion. And again, according to the authors, Nutmeg allows for flexibility in the input for conditions which we have a limited data range for, or large uncertainties, such as extraterrestrial environments or moons that we haven't visited yet and haven't gathered data on yet. And that's perfect for this project and what we want to do, because there are a lot of parameters of Europa that we don't have certain values for. We have only theoretical models or guesses for. So a little bit on how Nutmeg is structured. On the left side is a plot of essentially the idea behind Nutmeg, and on the right, there's a plot that I made showing the structure of how the simulations are built. So on the left side, you start with an energy source and a nutrient source defined by the user. On the energy side, that epsilon sub m refers to a microbial maintenance efficiency that is related to any process that uses energy that is not directly related or contribute to the growth of the organism. That epsilon sub ut is the nutrient uptake efficiency, which represents the effect of limited availability of carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur on the amount of biomass that the organism can actually take in per unit time. And then once those different parameters and those different processes are taken care of, any extra nutrients or energy goes toward the growth of the organism or the population. And then quickly on the right side, we have the structure of the simulation. You start with a base organism, which again, the user defines, and the environment, which holds basic properties of the environment like temperature, pressure, volume, that sort of thing. You choose your population type, either a colony or a horde. And for now, I'm going to skip the definitions between those two just for sake of time. But you choose your population type, those go into a culture, and then you combine that with your environment, and you also define your reagents and your reactions in that environment, which go into the reactor object. You combine your culture and reactor into your ecosystem and simulate that over a set amount of time, again, defined by the user. And at the end, you can get many different outputs, which I'll be showing a few here about the energy available in the environment, the amount of biomass it can support, the evolution of the biomass over time, methane produced specifically in this case, and many other outputs. So, and I'm going to be going over my preliminary results so far. And one thing about these results is for all the results I'm going to be showing, the parameters for Europa were used in place of those for Enceladus. The reason I say that is because the code comes with an example file for running these simulations for Enceladus. And so far, we've essentially been editing that Enceladus example to include the parameters for Europa instead. All the temperature and pH ranges for Europa are based on other state-of-the-art models. And we are also assuming a typical optimal methanogen, or a TOM, which is an artificial methanogen designed to behave as if any given temperature is its optimal temperature. And it'll mimic the growth behavior of a real methanogen, which is well adapted to that said temperature. And then also, lastly, the plots you'll be seeing are produced using Nutmeg's built-in or default plotting code, but again, using the parameters and properties of Europa that we have so far. So first is this plot here that shows the available power supply to these methanogens for different values of seawater temperature and bulk ocean pH. The orange line you see there refers to the maximum temperature limit for life. Also, the range of temperatures and range of pH here are the ranges that we have for Europa so far based on those models that I mentioned in the previous slide. And as you can see here, what we see is as the temperature rises, the power supply also increases, but there's really no change of pH, which is a little bit weird because you would expect a change for pH. And again, these are very preliminary results, and we are currently in the process of making a more accurate Europa environment file, which may change this result in the future. Next, we have this, again, a power supply available, showing the power supply available for the environment. However, this is more specific combinations of temperature and pH, or keeping one constant and varying temperature, or keeping temperature constant and varying pH. Also, this plot is broken up. On the left side, that's the top of the plot, and the right side, that's the bottom of the plot. But again, here you can see with changes in temperature, there is an increase in the power supply available, but again, there's no change or very, very little change in the power supply with any sort of changes in pH. Then we have this. This plot shows the criteria for habitability based on different temperature and pH values. Again, this left side of the plot is the top of the plot. The right side is the bottom, and to be split due to space reasons. But the one panel of this that I would like to highlight is the very left side with the darker orange color. That plot is showing the criteria habitability to exceed maximum optimal maintenance, and also to show whether or not exponential growth of the methanogens is possible based on these combinations of temperature and pH. And you can see here, based on that color, for the dark orange rectangles you see filled in, there is enough power available with those temperature and pH combinations to allow for exponential growth of the methanogens in Europa's environment. We then have a couple plots looking at growth curves. Here we have the amount of biomass in cells per liter over a range of different temperatures and a range of different nutrient limiting scenarios. So going from left to right, we have the limiting cases for concentration of CO2, H2 gas, phosphorus, and then the phosphorus uptake rate. The different colors here correspond to the optimal -1%, -10%, and -40% of the optimal concentration. And then similarly, slower and slower rates for the phosphorus uptake rate in the last column there. And the filled in regions here are various yields of ATP per mole of CO2 ranging from 0.5 to 1.5, and the solid lines are at a 1.0 yield as well. And what's interesting here, as you can see at the higher temperature at the bottom, these lines just end. Our current theory of why this is happening is potentially the methanogens just can't exist past a certain point in that time frame, aka the population will die out. But we're still looking into why that is stopping instead of continuing on like the other plots. But we think the population just can't survive at those temperatures. Here are some other various growth results, again, for the same nutrient limiting cases. But this time we're looking at the amount of methane produced at the very bottom. Then in the middle, we have the maximum biomass after a set amount of time. And then the peak growth rate, again, after a set amount of time of simulation. The x-axis there, you can see, is a range of different temperatures. And this is, again, after the simulation has run at a given temperature, this is the maximum CH4 produced biomass and peak growth rates. And what's interesting to note here is in the peak growth rates at higher temperatures, you can see that, or not higher temperatures, but at, again, optimal CO2 or -1% CO2 as expected, that growth rate will increase very high and then drop off very rapidly at higher temperatures. However, the CH4 produced seems to increase in a couple cases at those higher temperatures, even though the population seems to die out or stop growing at those higher temperatures. And then lastly, I want to go over some of the summary and future work we plan on doing. So like I said, the central theme of this project is constraining the habitability and ecosystem properties of Europa and eventually other worlds. And this approach can help also guide the design of future observations and data collection missions, as well as help analyze and interpret the data coming from such missions such as Europa Clipper or other various laboratory experiments that will be done and or field studies that will be done in the future as well. As I showed, these preliminary results and other experiments show that the environment for Europa allows for methanogens to survive and potentially exponentially grow from that orange plot as we saw earlier. And then future simulations will provide a more insight into the habitability of Europa and other worlds, and again, provide a point of comparison with future collected data. Some of the future work we plan on doing is to build a more accurate environment for Europa, which I'm currently in the process of doing, as I mentioned earlier. This will look at more properties of Europa besides temperature and pH variations to create a more complete picture of the ocean and run these simulations again. We also plan to expand to other worlds and other moons, namely the Hycean worlds, which have a water ocean underneath a very thick hydrogen-rich atmosphere. And we also have plans to look at the Hadean Archean Earth, as the ocean and the atmosphere was much different back then, namely a little bit higher pH, a little higher temperature. We also want to combine this model with other models to create a deeper, or to perform a deeper, more broad analysis of the European environment. And we also have the idea of adding in additional modules, including the motility of organisms, which would act as another energy sink, potentially leading to a lower final levels of biomass for the same input energy or nutrient input as well. And that's about it. Thank you very much.